The central dogma of biology describes the process of transcription and translation by which information in genes flow from DNA to RNA to proteins. This was a phrase coined by Francis Crick in 1958, where information passes from DNA to proteins through RNA. However, proteins cannot send the information in the opposite direction. DNA stands for deoxyribonucleic acid. It is made up of smaller molecules known as nucleotides. Nucleotides are composed of a sugar phosphate backbone with nitrogenous bases that pair to other specific nitrogenous bases. DNA is the genetic code that all living beings have and is responsible for the differences between organisms. This genetic information is stored in the nucleus of eukaryotic cells and has a double helix structure. It codes for the proteins and traits of an organism, which correlates to how genes are expressed in an organism. The most accurate representation of a gene expression is that one gene codes for one RNA molecule, which has the potential to be translated into an amino acid chain. RNA stands for ribonucleic acid, and it serves many functions when it comes to coding for proteins. Unlike DNA, RNA is a single-stranded molecule that can be located outside of the nucleus.
The first process is transcription, which takes place in the nucleus of eukaryotic cells. However, in bacteria, this takes place in the cytoplasm. Because their DNA is not bound by a nucleus, the first step of this process is initiation. In initiation, the DNA in both prokaryotes and eukaryotes are separated by an enzyme called helicase to start the process in creating mRNA. In bacteria, RNA polymerase binds directly to the start of the gene sequence, or the promoter, while in eukaryotes there must be a specific DNA sequence known as the top box located on the template strand. This is represented by the nitrogenous bases thymine and adenine in the sequence TA. The polymerase located the top box because thymine and adenine have less bonds than guanine and cytosine bonds, so it's easier to break. Once the sequence is recognized, RNA polymerase can bind to the promoter, and helicase separates the DNA strands by breaking the hydrogen bonds. RNA polymerase then transcribes the DNA template strand into messenger RNA, or mRNA, by adding nucleotides. This causes elongation, which is the second step of this process. The mRNA strand moves in the 5' to 3' direction by adding nucleotides to the strand. When RNA transcribes DNA, it replaces thymine T with uracil U, which binds the adenine A. The last step is termination. This occurs when RNA polymerase transcribes a specific terminator sequence from the DNA template strand. This causes polymerase to detach from the DNA. It will also get a 5' cap and a poly A tail. The poly A tail consists of numerous adenines, which will protect the mRNA as it is exported from the nucleus. Once the sequence of target DNA is transcribed into mRNA, it is ready to be used for translation in prokaryotic cells. However, in eukaryotes, at this point the product is referred to as pre-mRNA, and it still has to be processed to reach its final form. Part of pre-mRNA processing includes the splicing of introns and the joining of exons. This is known as SNRPs, or snRNPs. Introns, or non-coding sections of pre-mRNA, are cut out, and the exons, which are coding sections, are joined together. SNRPs help cut the introns and join the exons. Bacteria and other prokaryotes do not have any introns, allowing for these types of organisms to skip the process of refining mRNA. The information can now be carried to the parts of the cell responsible for the translation of genetic code into proteins.
The next process in Central Dogma is translation. Translation is similar to transcription. They both have 3 steps: initiation, elongation, and termination. But in translation, the mRNA goes to the ribosome, which are located in the cytoplasm. And tRNA, or transfer RNA, brings amino acids to the corresponding codons to make a protein. The ribosome contains 3 sites, in A, P, and E. The A site is where incoming tRNA bring their amino acids. The P site is where the growing polypeptide is located. And the E site is where the tRNA exits the ribosome. tRNA acts as the interpreter of the message delivered by the mRNA. It's transcribed in the nucleus and will pair its anticodons with the mRNA codons. A codon is a series of 3 nucleotides that are used to make an amino acid, which is used to make a polypeptide. During initiation, the mRNA attaches to the ribosome and the initial tRNA goes to the P site with the anticodon UAC and binds to the AUG nucleotides. AUG is the start codon, which starts the whole translation process. After the initial tRNA binds the anticodon with the codon, elongation begins.
During elongation, a new tRNA is brought to the empty A site, and the tRNA in the P site transfers its amino acid to the tRNA in the A site. An enzyme named peptidyltransferase helps transfer the amino acid from one tRNA to another. The ribosome then moves down the mRNA, and the tRNAs are transferred to the next site. The tRNA in the P site is moved to the E site, where it exits the ribosome to get another amino acid from the cell. And the tRNA in the A site is moved to the P site with its amino acid, and a new tRNA is transferred to the A site. The whole process continues, and during the synthesis of the protein begins to fold and coil. Chaperonin is a protein that helps ensure the polypeptide chain folds correctly. This entire process is referred to as translocation, the full process of entering and exiting the ribosome. The whole process will eventually come to an end when the codon UAG, UAA, or UGA is located. Once it is located, no more amino acids are added, and the tRNA along with the polypeptide are released at the E site, and the ribosome breaks. The polypeptide, also known as protein, is then carried to its appropriate location, and the mRNA will eventually break down.